Hello, and thank you for joining us today for this webinar with BioChain Institute and Enable Medicine, presented by Samba Scientific. My name is Jason Amsbaugh, and I'll be the moderator today. In this webinar, Dr. Rikita Gakhar, an application scientist from BioChain Institute, and Dr. Gulpreet Kaur, Enable Medicine's Business Development Director, will present data on the Enable Medicine computational pipeline and how it's advancing the power of spatial transcriptomics in translational research. I'll go ahead and turn our time over to Drs. Gakhar and Kaur.
Hello, everyone. I am Rikita Gakhar, and for today's presentation, I will be talking about a case study on 10x Visium spatial whole transcriptome analysis of differential expression for biomarker discovery in colorectal cancer. To begin, I would like to introduce BioChain. BioChain was established in 1994 in the Bay Area, and since then, we have been providing biospecimens and related services for over 29 years. And more recently, in 2021, we also started providing spatial biology services, which will be the focus of today's presentation. But to give you an overview of the major categories of services and products that we provide, here are the 6 major categories that you see on the slide. And I won't be touching upon the others since the talk today is focused on spatial biology, but please feel free to visit us at biochain.com or email at info@biochain.com if you're interested in learning about any of the other products and services. So let's delve deeper into spatial biology services that we offer. At present, we are offering 10x Visium direct placement as well as site assist services, besides other platforms such as NanoString, GeoMx DSP, SlideSeq from Curio Biosciences, and Codex in partnership with Enable Medicine. So let's take a step back and look into why we need spatial biology. And it all began in the early 2000s with the development of next generation sequencing technologies. Bulk RNA sequencing became a popular genomics tool to classify cancers, to study or discover gene fusions and biomarkers, as well as disease diagnosis and optimizing treatments. But however, with bulk RNA sequencing, it's kind of like the smoothie on the left, the fruit smoothie that you see on the left side of your slide, where you lose the context of individual cells, you know, like the one you see in the middle, which is a fruit salad, in which you can see individual fruit pieces. So single cell RNA sequencing technologies were developed to study individual cells. And with single cell technologies, we are able to identify and trace back the transcripts to individual cells, like in the case of this fruit salad in the middle. However, we lose the context of location and neighborhoods. And tumors in biology in general is very complex, and tumors are interacting spatially and temporally with their tumor microenvironment, which includes not only tumor cells, other tumor cells, but also immune cells and other cells. So there's highly complex spatial and temporal communication going on all the time between these cells. So spatial context becomes very important to study these diseases. And kind of like this case in this nicely arranged fruit platter, where we can see where each individual fruit piece is and what are its neighbors. So then moving on, herein spatial biology becomes very important, since now we don't have to study the histology separately from molecular biology. And spatial biology enables us to combine these two aspects of the tissue and study it in its entirety. Kind of like this example here, where you see the histology image on the left, and the gene expression data associated with this tissue section on the right. And in the middle is what you get from spatial biology, which is the molecular data overlaid on the tissue, the histology image. So now you can tell which gene clusters are where in the tissue section. And that helps us not only characterize the tumor microenvironment and study cell-to-cell interaction, but also to predict patterns of evolution and heterogeneity in these complex diseases, and enhance the biomarker discovery so we can come up with early diagnostics and enhance therapeutic interventions. And so that brings me to my next question: why BioChain for your spatial biology experiments? So BioChain has an in-house biorepository, a tissue repository of IRB-approved donor-consented tissues. We, besides these retrospective collections of tissues, which include FFPE and frozen tissues, as well as matched pairs, we also provide prospective collections and have well-established protocols. Besides that, we have histopathology lab, which allows us with the flexibility to use customer-supplied material, and we can construct TMA from that material, FFPE blocks, OCD blocks, and carry out the whole entire workflow for spatial platforms all the way up to sequencing. And then we have partnership with Enable Medicine, which can help you with in-depth data analysis, as well as we offer multiple platforms to fit the needs of your project, to customize your project and answer your research questions in the best possible way. So let's take a look at the 10x Visium workflow. So 10x Visium offers two different workflows. There's direct placement workflow for frozen as well as FFPE tissues, and then the site assist workflow for FFPE, frozen, and archived tissues. Here I'm showing an overview of direct placement workflows for both FFPE and fresh frozen samples. For direct placement, 10x offers the Visium slide with 4 capture areas. Each capture area is a 6.5 mm square and is lined with a lawn of over 5,000 spots. And each of these spots have spatially barcoded probes. So we can carry out the workflow. And the only difference in the beginning of the fresh frozen workflow from FFPE would be the optimization for permeabilization time that is required with the fresh frozen tissue. And then we can carry out, once the tissue sections are placed on the Visium capture area, we can carry out staining and imaging, probe hybridization, and so on, all the way up to library construction and sequencing. So then looking into site assist workflow, the site assist Visium slide comes with only 2 capture areas instead of 4. However, it comes with 2 different size options, the smaller one being 6.5 mm and the bigger one being 11 mm, which gives us the option to accommodate for larger tissues. And site assist workflow is much similar to the FFPE workflow, except the beginning few steps can be carried out on standard glass slides, which also allows us to section the tissue on standard glass slides or use pre-sectioned tissues. And site assist literally just assists us to transfer the probes from the tissue section onto the Visium slide. So with that, I would like to go into case study. And so at BioChain, we used some of our in-house repository tissues that for 10x Visium characterization, and all these tissues can be found on our website for purchase along with the data. But for today's presentation, I will be talking about CRCs, colorectal cancer in particular, for which we studied 4 different donors, tumor, as well as just normal tissues. So why CRC? CRCs are highly diverse tumors. CRC is the 3rd leading cancer diagnosed globally, and about 1.2 million cases are reported every year. And more recently, the trends have been shifting towards increased incident in adolescents and young adults. And given the diversity, the molecular molecular diversity in these cancers in early stage and early onset and late onset, also as well as hereditary and sporadic cancers, the treatment fashion still remains the same for both early onset and late onset. So there is definitely a need for more in-depth knowledge of these cancers, classification of these cancers, so that we can do patient stratification and early interventions, early therapeutic interventions. So let's take a look at the donors that we selected. We selected 4 donors. This is the clinical data of these 4 donors. We took tumor as well as normal tissue from each of these donors and did DV200 score for all of them. The threshold for direct placement for DV200 score is above 50%. So all these tissues were above 50% DV200 score. And also these tissues were screened by our pathologist. So here is an example of a web summary of one of these tumor samples. This is something you can find on our website as well for these characterized samples. So here, this web summary is showing you the tissue section on top of one Visium capture area and the number of spots that were covered under the tissue, as well as median reads per spot and median genes that were detected per spot. And here is a t-SNE plot for clustering. So in summary, we were able to detect about 16,000 genes in each sample, and the median of about 5,500 genes in the tumor section per spot, and 1,600 genes per spot in the normal section. So we took this data and used 10x's free software, the Space Ranger, to do some preliminary analysis to compare the tumor tissues with the normal tissues, vis-a-vis CRC biomarkers, as well as immune cell biomarkers. So let's take a look at the tissue sections. So on the left here, you see the tissue section annotated by our pathologists. You can see mucosa, submucosa, and muscularis layers in this normal tissue section, colon section. And on the right here, you see the annotation overlaid with clustering data. And as you can see, the clustering correlates very well with the pathologist's annotations. And here is a slide for showing the annotations and clustering analysis for the tumor tissues. And we found similar results with IHC as well. Those results are not shown here, though. So moving on, we constructed some heat maps to compare gene expression between the tumor tissues and normal tissues. And we found specifically these markers expressing in the tumor tissue compared to normal tissue. We also constructed violin plots for these 5 particular CRC biomarkers, and as expected, found these overexpressed in tumor tissue sections. Similarly, immune B cell markers were also overexpressed in tumors as expected. And to summarize the key findings here, 10x Visium data corresponds very well with both H&E and IHC annotations, which is very important pathologically. And it provides immense transcription data in spatial context of the tissue, as well as it shows a lot of potential for early biomarker discovery, exploration of tumor microenvironment, and further validation studies. So for future roadmap, in the next 6 months, we are looking forward to doing a lot of multimodal projects, combining 2 or 3 different platforms and yielding even more comprehensive results, as well as we are onboarding proteomics workflow, and we are very excited to onboard Visium Institute analyzer as well. Shown here is a mouse slide analyzed by Visium Institute analyzer. So really excited for this future roadmap. And to conclude, I would leave you with a few highlights of our key strengths. We have a biorepository, which is IRB approved and also has rare tissues. We have the flexibility of using customer supplied material, experience with tissue handling and TMA construction, and in-house pathologists and molecular biologists. And we also have pre-characterized tissue sections available for purchase with the data. And you can check these out using this QR code on our spatial biology webpage, as well as multiple spatial biology platforms for you to choose from to best answer your research questions. With that, I would like to thank you all and pass it back to Jason. Thank you.
Thank you, Rikita. Our next speaker, Gulpreet Gakhar from Enable Medicine.
Hello, everyone. Can you hear me?
Yep, sounds great.
Sounds good. All right. Good morning, everyone. My name is Gulpreet. I'm a business development director at Enable Medicine. My PhD is in cell and molecular biology from University of Wisconsin-Madison, and I'm really enthusiastic about working alongside the scientific community to introduce our innovative technologies that are going to hopefully help further scientific progress. On the webinar today with me, I have Steve Hamel, who has supported generation of the data that I'll be showing you today, and will also be helping us answer some of the questions. All right, so at Enable Medicine, we are a team of systems immunologists, engineers, bioinformaticians, and life scientists like myself. We are working together with a goal to solve critical problems for drug developers using AI and human-driven insights in biology and medicine. We are utilizing and developing cutting-edge technologies that will allow us to extract as much meaningful information as possible from a data set and unlock the full value of scientific data. One of the problems that drug developers face is how to improve outcomes for clinical trials. And one way to improve these outcomes is to use targeted therapies by using pre-selection biomarkers. This graph here, are you able to see the graph? Okay, so clearly we're having some issues here because I can't see these graphs. Okay, but the idea was to show you the phase transitions of clinical trials from phase 2 to phase 3 over a period of 10 years. The bar graph that I hope you can imagine here actually shows this data. So this is obtained from this report called Clinical Development Success Rate that's mentioned here. And the report also actually discusses this thing called the likelihood of approval of a clinical trial, of a drug in a clinical trial. And that's a compounded probability calculation based on success rates of trials. So this report actually showed that the programs with trials employing these patient pre-selection biomarkers have a twofold higher likelihoods of approval that is driven by primarily the phase 2 success rate. So this data was collected over 2011 to 2020. So the biomarkers that are included here range from histological, physiological, and molecular biomarkers. And we're now in the age where we can combine these with spatially resolved technologies to obtain deeper answers. And at Enable Medicine, we are using these spatial biomarkers with the goal to improve outcomes for clinical trials. So what can we do with just biomarker discovery? One, we can explore biomarkers for a given indication or to evaluate therapeutic efficacy. Two, we can use these biomarkers and lead them and interrogate them to help us understand the mechanism of action a little bit better. This finally allows us to build more AI-based models that allow us to predict clinical outcomes better based on our understanding of the underlying biology. And finally, this helps drug developers stratify patients based on clinical outcomes and select the expected responders for a clinical trial, for example. So we work with various pharma and academic partners to provide complete solutions for translational and clinical research. It starts with exploring and identifying the key questions and building an experimental plan. We then figure out what data needs to be collected, right? For spatial proteomics, we do hyperplex imaging in-house. We have a panel of about 51 to 100 proteins that span immune, tumor, or inflammatory markers. For spatial transcriptomics, we use our partner networks such as BioChain to generate the data. And then moving on to data analysis, we've built all of our analysis pipelines in-house primarily. We have pipelines built for spatial proteomic data that are actually available to academic users for free on our no-code platform that I'll talk about at the end. What I'll focus on today is our spatial transcriptomic pipeline and how to integrate that to the spatial proteomic pipeline. And finally, we don't stop at simply analyzing the data and turning that over. We actually extract insights and present key themes using our expertise from our scientists and also from our AI-based algorithms. All right, so these are the types of data we can handle. So, H&E, IHC, and then on the imaging side, we can handle most available data types, which includes low or hyperplex immunofluorescence like COMET or CODEX, metal ion-based assays such as MIBI or IMC, and even FISH. And on the transcriptomic side, we can handle spatial transcriptomic data, bulk, single cell, ATAC-seq, whatever we need to do, whatever can actually give us good information. The most important part, though, is how we analyze these data. Most of the technologies operate at a single level, and from them, we can identify not just various cell types, but also explore the interactions between cells and neighborhoods of cells. This allows us to explore complete biological networks, right? And this gives us confidence in our predictive models. And finally, we can use all of this information to extract various spatial features that are of interest to various researchers. So why are we focusing on spatial biology here? Here's a graph that was published in Bioinformatics this year, comparing the concordance of various algorithms with clinical outcomes of patients. In orange here are non-spatial features that are commonly being used, just average of a biomarker across an image or in cells, for example. And in the gray here are simple comparison methods such as protein positivity values or protein interaction counts. Now, let's look at the green bar here, which is showing neighborhood matrix. It takes into account not just the cell type proportions, but also the cell types that are surrounding each cell. And as an example, it's the difference between asking, are there cytotoxic T cells in the tissue versus are the cytotoxic T cells near the tumor? Are there Tregs surrounding this tissue area that may be suppressing the immune function? So these parameters are relevant to why a patient would respond to a given treatment or not. And I hope this can illustrate why the neighborhood matrix has such a high concordance value with clinical outcomes. Now, going one step further, now notice the blue bar. This is the combined concordance index of two different spatial technologies, the neighborhood matrix and the Ripley's K. It's a transformation of the Ripley's K function, which is a measure of the pairwise distance distribution of different points in a pattern. Now, this combined approach gives us more data and more confidence in our results. And today, I'm going to show you an example of a similar multi-omic approach to extract biomarkers. All right, so this is data that we published it's a paper that's recently published in bioRxiv. So here we're just assessing the spatial organization of a breast cancer tissue using Visium and CODEX approaches and integrating the two. All right, so from CODEX, this is, the new name is Akoya phenocycler. So this is hyperplex staining. So from this, we were able to identify different cell types, different interactions, and different neighborhoods that are altered or are of relevance in this given tissue. So we interrogated it with about 51 proteins at single cell resolution and just to identify the different functional neighborhoods. And on the transcriptomic side, we try to figure out the transcriptomic profile of the tissue, right? And understand which genes are up or down regulated. And if we had full cohorts, we could go in and identify what's happening in the different cohorts over here, right? So, and then correlate the two. Okay, so the goal here was to understand, even within the one tissue that we have, are we able to identify different, or are there different transcriptional states in the various neighborhoods that we've identified in the one breast cancer tissue? So identify neighborhoods in the using codex, and then going in and seeing what transcriptional variations exist or don't exist in these neighborhoods. All right. So I'm going to start by talking us through just the transcriptomic pipeline that we used and focus a little bit more on that. And then I'll show how we did the integration. So Visium, 10x Visium is what we use for this data set. And it's a spatial transcriptomic assay. It measures the mRNA abundance of a tissue section, but it maintains the spatial resolution. So each of these is a spot that you can see that we can measure mRNA expression, mRNA abundance from. But each of these spots actually covers about an area of 55 micrometers. So that covers about 2 to 5 cells. So as you can see over here, each spot will have multiple different genes and different mRNA abundance values, which correlate with expression, roughly. So this is a general pipeline for spatial biology, right? We use a very similar pipeline for proteomics and for transcriptomics, but I'm going to focus more on the transcriptomic pipeline over here. So you start with tissue segmentation. You go into your quality control and batch corrections. If you have batches, then you obviously want to make sure you're doing that. And then you cluster these cells. So then you go in and group these cells into various cohorts. Well, not cohorts, group these. The goal is to identify different spots or different clusters of cells. So that's a clustering part, then you go in and phenotype what each of these clusters are. And then we go in and integrate with multiomic data. And then if we have cohorts, we can compare what changed between the different cohorts. So let's start with the first one. We'll start with tissue segmentation here. Okay, so for H&E, for Visium data, we actually just took the H&E image of the tissue and used the standard Space Ranger algorithm to do the segmentation. And then on the other side for Codex, we actually used our DAPI image from the Codex to do the segmentation using Deep Cell. And the idea is to identify where your cells are, right? So is to identify the precise boundary of every single cell that you can identify. And at Codex, with the single cell technology, with a single cell resolution, that's possible. Here we're identifying basically as much as we can using the H&E image, right? And then we go in and we visually mark the areas of damaged tissue or folded tissue. So you do want to exclude some of these regions. And I'm going to show you an example of that over here. So this is not the tissue that we use, but just an example. So you can see that these spots line up perfectly over here, but these spots would need to be excluded. And you also start to see low counts in areas of folded tissue. So a couple of steps we do to do this quality check is first just visually inspect the tissue to make sure that the tissue quality is good. The next step is to filter out spots based on UMIs, right? So the total RNA counts per spot. So for example, if you have less than 50 total RNAs in a spot, that might be an indicator of damaged tissue and you might need to remove those. If a tissue hasn't fully attached to the probes, you also get too little mRNA, and that might need to be removed also. There are instances where you might need to remove a spot because it has too many RNAs, but I would be really careful with that, and you would want to check to make sure whether it's true biological representation or not, right? And the same way, the another step would be to filter out based on genes. So if a spot has maybe less than 10 genes, that might be a spot that you might want to remove because it's not representing really well. And finally, something that we consider is a percentage of a spot's total gene expression that might be due to mitochondrial genes or platelet genes. And we have different in-house parameters for that. So for example, if the mitochondrial genes appears to be over 15% of a cell state, we might remove that. If platelet genes appear to be more than 30% of a cell state, we might remove that. And this is just to account for the biology and the kinds of RNAs that we want to see in our actual assay represented. Okay, so I walked us through just the spot filtering and the spot filtering method, but here's the example actually from the tissue that we used in the paper that we've published. So these are the counts here before QC filtering, and then we built a histogram to show the total RNA counts per spot, and then we removed anything that was less than X of 10. And you can see over here that the red spots are the spots that were actually removed and the green spots were kept. And then we went into gene filtering. In gene filtering, you always want to remove genes that are technically deprecated genes by assess using pseudogene or by gene cards. And actually, it was over here that we removed anything that was less than X equals 10. So removing any genes that are expressed in less than 10 spots. And this is, again, to get to a point where you are getting good expression, right? And true biological representation. Okay, so having completed that initial tissue segmentation and quality control assessment, if you have multiple cohorts here, this is where you would apply some batch corrections also. It would be, it's pretty standard to do like a normalization or a batch integration workflow. Which is, which has some potential here. But the next steps are to cluster and phenotype these cohorts, these tissues. All right, so let's talk about that. So this is our method. I know there's many different methods out there to do clustering. This is the method that we commonly use and recommend. So in this, we're essentially classifying the spots into different neighborhood types, right? And hoping for better understanding of each of these neighborhoods in the tissue. We start by doing a dimensional reduction. Data is actually, even before that, data is transformed by normalizing the expression of spots relative to total count using a log normalization. And then we find the most highly variable genes in the data. And then on that, we do the dimensional reduction on the top most, maybe 2,000 most highly variable genes. From there, we're applying a nearest neighbor algorithm. So that means for every cell that is annotated in the data set, the algorithm enumerates the cell types of its k nearest spatial neighbors in Euclidean space, right? So the k nearest neighbors approach is essentially just dividing the data into rough cohorts. And here we actually, you do have to tell it how many cohorts you want. So here we started with a k of 50. And then this data, once you've done that initial, nearest neighbor calculation, this data is then fed into the lead-in clustering. So lead-in clustering is a community detection approach that identifies the data into various clusters based on the interconnectedness. So based on that, we got our final clusters, and they're labeled 0 to 6 over here. And now you want to understand what's in these clusters, right? What are the key themes that are coming up? So here is a heat map that's showing the genes in a given neighborhood. The yellow genes are upregulated and the pink genes are downregulated. If you take cluster 1 here, for example, and compare the data, the total data set compare the genes in cluster 1 to the whole data set, we end up with a differential expression plot over here, which is what's showing, which is what we're showing over here. You can start to look at what genes are represented in the pathway. The ACTA and ACTG genes, for example, are actin-related genes, and fibronectin genes are also represented. So from here, you would do a gene ontology analysis to really identify the relevant pathways in this neighborhood. Okay, all right. So from that point on, we actually went in and tried to do a multi-omic integration, right? Because we want to understand what's going on over here now. Okay, so we used our approach called EM-Object. So this paper was recently released at bioRxiv earlier this year. And I want to give you a general sense of how we think about the data in this. There are obviously many different aspects to EM-Object. So EM-Object is a data abstraction method, right, for spatial omics. So essentially what's happening is if these were all of the different layers in your data, the EM-Object will organize around a codon matrix and treat that as a base layer and use all your other layers and attributes as attributes, right, and add all of that. So what's going on over here is that you're able to layer your Visium data onto your Codex data or your Codex data onto your Visium data. And it's agnostic to data type. So you can analyze Visium, maybe Codex, GeoMx data sets, and integrate all of these very easily. So having built this, this is what we did with this. So here's the tissue that was imaged with Codex. And these are the Visium spots that were identified using the method that we just talked about. And finally, we went in and tried to align this data set, right, using EM-Object slice function and layer-specific segmentation masks. All right. Okay, now I want to show you the results that we pulled out from this integration of just this one tissue. What could be found with just one tissue? Right. So here are the cells that we identified with Codex. And so this was single cells, so we were able to identify individual cells. And from there, we went in and identified the various neighborhoods using Codex. And remember, we had also identified the neighborhoods using Visium. So now we start to integrate these and come up with Codex Visium alignment and identify the final clusters that we would come up with with this Codex Visium aligned data. All right, now let's start looking at the transcriptional profiles of each of these neighborhoods. Now that we've identified, NT0, so neighborhood 0 to neighborhood 5, we're looking at 6 neighborhoods over here. These are the various transcriptional profiles that we can pull off, that we can pull out in each of these. And if you look at NT5, for example, because even visually, I think it's quite prominent how different NT5 is, even within itself. So you start to see that a major chunk of NT5 is V0 and a major chunk of it is V6, right? So NT5 actually had primarily ASMA positive, protoplanin positive, a neighborhood like cell type. So it's primarily a stromal neighborhood. And what we're showing here is that even that one neighborhood exists in distinct transcriptional states, right? So having investigated the marker genes V0 versus V6 within this neighborhood, we saw that V0 had marker genes that were associated with just tissue remodeling, cell adhesion responses, right? And then V6 contains marker genes that are associated with the immune system. So both V0 and V6 contain marker genes that were indicative of these being stromal fibroblasts, right? And these are consistent with the observed morphology in NT5. But the whole data, like when you put it together, it does suggest that there are similarly composed cellular neighborhoods that are, defined by proteomics, like NT5, but these can also exist in transcriptional states, which would not have been detected by just a unimodal analysis with just either codex or with just transcriptomics. So finally, just wanted to close this off by saying what we did in this study. So we use spatial transcriptomics to identify the transcription state of the tissue, right? We use spatial proteomics to identify single cells and the cellular composition and the cellular neighborhood, cellular niches that were present in the tissue. And then we integrate the two to identify distinct transcriptional states within the cellular neighborhoods. And this is the kind of work we are excited by. This is what we're doing every single day, right? We're interrogating patient data, clinical data, to explore these spatial profiles and extract strong biomarkers that could help enhance understanding of disease and clinical outcomes. All right. These are a list of solutions that we offer, right? We partner with pharma and academic clients to, just do biomarker discovery and patient stratification type of work. So happy to talk to you about more of these. Finally, I just want to quickly talk about the products we offer. So we've got the Enable Lab, right, that does the data generation and the spatial analysis for any translational clinical research. We have our Atlas, which is an annotated database that consists of multimodal data sets, right, that you can search across with, some of these have different biological conditions. So we're building that, and we're very excited about that too. And finally, we have a Enable Cloud platform that actually has no-code workflows for spatial proteomics. So the entire codex data set that we showed, you can do this on the platform in a no-code fashion. And then we also have custom algorithms that are available using our advanced module that can be integrated also with the platform. Finally, just want to leave by saying we have about 5 years of experience. We've studied about 20 plus diseases and profiled 400 million cells. These are some of our recent publications and some of our collaborations, academic collaborations. But we continue to partner with a lot of pharma companies also and support clinical trials or just preclinical research for them also. All right, that's the end of my talk, and I will stop sharing now. Thank you.
Thank you very much, Gulpreet. All right, with that, we're on to our Q&A session. So thank you to our speakers, Rikita and Gulpreet, for your presentations. I'd also like to introduce Vidya Sundaram, Vice President of Business Development at BioChain, who will also be answering questions. All right, we do have some questions. Some of the first ones. Can I study plant tissues with Visium?
Hey, Jason. Hey, Gulpreet and Rikita. That was a wonderful webinar explaining from the spatial capacities at BioChain and also how the analysis is done at Enable. Yes, to answer your question, Jason, we can do plants, and Visium has this frozen platform manually that we can use to place the samples, which is oligo-DT based, and that is a platform that can be used for any type of organism. It's tissue or organism agnostic. So we could use plants, insects, any other organism. And also the other advantage of using the frozen manual placement is that it does long reads, which is, I think, has its own unique advantages versus FFPE tissues, which is mostly, broken down RNA. So yes, it can be done. There might be some challenges, but yes, it can be done, and there are some publications.
Great, thank you. For a beginner in the field, what should one consider when designing a spatial project?
So For a spatial project, I think the first thing that people need to understand is what is the question they're asking. Is it a discovery-based question? Which means then we have the Visium is a great platform because you're looking at, in a slide, you have around 5,000 spots, and each spot has around 5 to 10. It can go up to 5 to 10 cells. Each spot is around 55 microns. There are, of course, some, gaps between the spots, but you're looking at the whole transcriptomics, and you can really get, the big picture, spatially understand the big picture. So it really depends on the type of question. And we see a lot of people coming to us saying that I really want to look at some, tissues or patient tissues. I really don't know what is going on, responders versus non-responders. And I really want to look at something, globally, spatially. But if you have other questions in terms of, for example, Xenium is not a sequence-based, it's more in situ, which, then we have panels. The 10X is coming up with specific panels for breast cancer and other things that you already know. Either you have some information through single cell or through Visium, then you can go ahead and, have some targeted questions that can be done using either Xenium. And also we have NanoString too. NanoString GeoMx, which also gives really, great information when you're looking for regions of interest. So it depends on that question, what question. Once you figure that out, and then we can talk about platforms and then the pricing and also the type of samples that you're interested in, whether it is patient samples, FFPE or frozen, is it in a block, or do you have some archival samples? So that also helps us, kind of narrow down the platform, that can be used. Finally, I think it's a cost. If somebody is interested in spatial, the initial cost also can be sometimes a sticker shock, but the data that you get, the information that you get is just incredibly rich. And with the current platforms, pipelines that people are building, I think it could be very powerful.
Thank you. All right. So the question, the tissue type, and the cost, key things to think about.
And the question, yep.
The next question here, which assays or what assays can be used with the enabled platform?
Okay. We can analyze data from pretty much any different types of assays, right? We have no-code algorithms available on our spatial on our cloud platform that academic users can actually sign up for free and try using right now. So that spatial proteomic pipeline is completely ready and free, and the spatial transcriptomic pipeline is built, and the integration pipeline is available as a service right now. So we can do any of these.
Great, that sounds great. Do tissues need to be consecutive slices to be registered together?
Actually, it's obviously better to have consecutive slices, but you don't need them to be consecutive. We have used nearby slices to do codex and Visium integration. It's obviously, like, I want to put a caveat here that the data quality will be better as you get closer adjacent, but it does not have to be consecutive.
All right. The other question here, I guess, just one question around sort of integration. Can you integrate codex data with GeoMx?
Yes, and we actually have a case study that will be presented at SITC this year showing GeoMx and codex integration.
Another question about data integration. Can you integrate Curio data with Xenium data?
We can, yes, we can analyze any of the data modalities that's available out there, and use our EM object to integrate different data types. The questions that we ask will be different for different modality, right? TEDx Visium is more a discovery platform. GeoMx is more focused on specific markers. So the questions that we ask will be slightly different. But technically, yes, all of these are possible.
Excellent, thank you. Here's a question. Do you think it's possible to obtain good spatial transcriptomic data on small samples? And so I guess that's the first part. And the second part of the question is the size of a sample a limiting factor?
So in terms of size, are you talking about core needle biopsies? Yes, it can be done. We have done some TMAs with small cores. So it can be done with any type of samples, whether it is core needle or resection samples. I mean, like when you mean by size, I'm assuming the size of the sample, right? The actual physical size of the sample. Yes, it can be done.
Okay. And then the question, is it limiting? So essentially when you have small samples, it's like it still works, but are you limited by the data that you would get out of that?
Yes, in that sense, you're going to get the data based on whatever, how big the sample is, right? And if it is just a core needle biopsy or a fine needle biopsy, yes, it is going to be limiting versus you're taking a big section from a resected sample.
Okay, but technically okay.
Yeah.
Next question is, how many markers do you typically examine in Codex for FFPE tissue of, say, colon cancer or breast cancer tissue? So ultimately just a plex question here. How high is the multiplex and what do you guys normally do?
Yeah, we actually have a standard panel of 51 proteins. It's our immuno-oncology panel that has, immune markers, immune checkpoint markers, different myeloid cells, tumor cells, different tumor cell types. But it can also add custom markers, right? Like I think we have plenty of barcodes to add up to like 67 and even up to 100 biomarkers at this point.
Okay, thank you. Technical question. Can you do codex from tissues preserved in DNA, RNA shield or similar buffers?
I actually haven't tried, like I don't know like what DNA shield is. Sorry, but I can check on that and confirm that. But if it's like we have done archival tissues, right, with codex. So it's pretty hardy.
It sounds like likely yes, but maybe a specific question there to follow up on. Just one or two more questions here. We're just about out of time. This would be, I guess the first question is that pre-selection of patients in the graph that wasn't there results in a 2 times higher likelihood of clinical trial approval. So are you aware of any clinical trials currently where spatial biology has been used to pre-select for patients, or do you think that's more of a future state?
Well, we have clinical trials currently that we're working on and interrogating using spatial biology. So,
so the future is now, is what you're saying.
Definitely in the near future, yes.
All right. Very good. And then this next question looks like the last question that we have time for. It seems like there's a convergence of multiplex immunofluorescence and multiplex immunohistology or immunohistochemistry in terms of sort of clinical utility. How do you see spatial biology and multi-omic approaches starting to play a role in clinical testing in the next, say, 2 to 5 years?
I think that's a great question, Jason. And honestly, like the graph I showed already shows that spatial biology does have a higher correlation with clinical outcomes. So I think we're going to start seeing more and more convergence of these technologies with the clinic. I think Akoya, for example, is already working on a companion diagnostic. So there are companies moving in that space right now. And I think it's a very exciting time for spatial biology. We should see a lot of this coming up.
Yeah, I agree, Gul. Thank you, Jason. We are also working on multiple projects where, people are interested in multiomics, proteomics, and transcriptomics, and preclinical trial or early phase 1. We are getting requests, to do both. We are working on, trying to get some more, more details, and hopefully, this is where the future is.
Thank you. All right, well, that's all the time we have for questions. If we didn't get to your question, or if you have another question, feel free to reach out via email, or you can certainly visit our websites. Just a reminder, once again, this webinar was recorded. We will be sending it out when it's available for that. Stay tuned for more webinars to come and subscribe to our email list to stay up to date on drug discovery and development resources. Again, thank you to our speakers, Rikita and Gulpreet, NVIDIA, and everyone at BioChain and Enable Medicine for your time today. Thank you as well, attendees, for attending today's webinar. Understand that you're busy and appreciate your attention. Goodbye and take care. Bye-bye.